seminar with the title Novel Approaches in Quantitative and Structural Proteomics for Clinical Research and Diagnostics. So I'll give it over to you.
Thank you, Kiki. Can you hear me? Is it on? Thank you for the introduction and thank you for the nice day. Make me work the whole day. I thought it's like pretty relaxing. I don't know where the pointer is. Yeah. And yeah, before I start, I want to talk today about two aspects in proteomics: quantitative proteomics, which is right now pretty hot, and as well what I think is the next big thing in proteomics, structural proteomics. And I also think that we can use the two areas of proteomics for clinical research, and as well it goes sooner or later into diagnostics. But before I start, I want to give you a short impression about where I'm from, and I want to introduce it. Like I'm from Victoria, Vancouver Island, which is in British Columbia, and this is how it looks like, and even this is how it looks like actually right now. There's no snow, but it's very green because we have the Pacific rainforest, so lots of rain. And also, like actually, we took this photo like during the spring break, and what you can see is still very green, and also like no cars here on the parking lot, because after 3 to 4 months of rain in the week where you have like a few days off, like everyone is leaving the island and goes somewhere to Florida or California and see the sun. So it's not that cold, but a lot of rain. So we are at Genome Canada funded Science and Technology Innovation Center. There are right now only 5 centers which are funded by Genome Canada. 3 in genomics, and they are at McGill is one, then in Toronto and in Vancouver, and 1 in proteomics, this is in Victoria, and 1 in metabolomics, which is half in Victoria and other half is in Edmonton. So as a Genome Science and Technology Innovation Center, we are obligated to provide service in proteomics, which is in this area shown over here. We are engaged in research collaboration with Canadian academic, international academic, and more as well with industry. And we have a strong focus on technology development. And today I particularly want to talk about quantitative proteomics and structural proteomics. I want to start with quantitative proteomics. And we did, actually we do this for quite a long time. When I came over in 2006, I was quite excited about the MIM technology. And at the beginning, as usual, if you have like a crazy idea, a lot of people laugh at you, but it wasn't a stupid idea. Like we really were pushing the MIM technology, and in 2012, in December 2012, end of 2012, Nature Method called this approach as method of the year. It was fast. It was like quite, yeah, like it was good. And what exactly is this the targeted approach of this multi-reaction monitoring? So I made this figure when I gave a presentation in front of MDs, and it took me a whole day. Like it was always too complicated, came back. And so this is the final product. Multi-reaction monitoring is something that is actually not new at all. It's like around like for decades, and in the pharmaceutical industry that's being used for quantifying drugs. And now we can use this for quantifying proteins, but actually we're not analyzing proteins by itself. We're analyzing peptides, which you make, as you know, by proteolysis. And now we separate some kind of like a prefractionates through liquid chromatography, which is online coupled to the mass spectrometer. And the mass spectrometer, in most of the cases, is a triple quadrupole, where you have 3 quadrupoles in a row. And so the peptides coming in most of the cases are like ionized by electrospray and going into, since we have like if you analyze very complex mixtures like the blood proteome, the proteins in cell lysates and so on, it's a very complicated mixture. So you never have like a really baseline separation of these peptides. So they're coming, a bunch of them coming in like this. And the first quadrupole acts as like a mass filter. And so like we know exactly at this time this peptide is coming into the mass spectrometer, and we adjusting the voltage that only, I think this peptide is going through the second quadrupole, shown over here, and then it hits some gas molecule, falls apart, like this, and the third quadrupole acts like as well as a mass filter, then only the fragment, a certain fragment, finally goes to the detector and gives us an ion chromatogram as shown over here. And what I said is nothing really new. It's well established, this method in the small molecule quantitation area, but it has like extreme advantages. Particular, it is pretty much the most sensitive MS approach we have right now. And sensitivity is when you're analyzing clinical samples, or in general like biological samples, a very critical point. And we really advocate for the use of internal standards because internal standards, they have several advantages. One of them is really improving the specificity if you're using internal standards. I just mentioned that what the detector is finally giving us is an ion chromatogram. So not an MS spectrum, ion chromatogram. That means like this fragment is coming out either at 29 minutes or 30 minutes. We don't know, we get like two peaks here. But if you're using internal standard, which is the peptide, the same peptide what we're analyzing, at the same chemical structure, at the same sequence, however, is isotopically coded. It means like it's a little bit higher in mass. It's always the same old trick when you're using mass spectrometry, you're using isotopically coded or labeled molecules. And we are using N15 or C13 isotopes, and the reason is because they have no effect on the retention time. So they need to come up at the same time as the endogenous peptide is coming out. So then it's quite easy, like the blue one is our endogenous peptide, and the red one is our standard peptide, and they need to come up at the same time. So this one is really what we're looking for. This is what we want to measure. And also you can see that peak shape needs to be pretty much the same as what we have from endogenous peptide. And another advantage, and this was really a big breakthrough, like in proteomics, is then when you use these cis peptides, you improve your reproducibility. And we did this experiment where we analyzed 45 plasma proteins, and we analyzed it 12 times. And if you do this and if you don't have any internal standards, you're getting percent CV on average between 25 and 30%. This is absolutely not acceptable for the FDA. However, if you use internal standard, then you're pushing the percent CV more like in the 4 to 6% range as shown over here. And when you're not only giving the internal standards equivalent, that means like for all of these 45 plasma proteins, we're adding the peptides, the internal standard at the same amount. If you do it like in a more sophisticated way, it means you balance this out. If you like want to analyze albumin at a concentration of 40 milligrams per mil compared to something what is like in nanograms per mil, you're give more internal standard to the albumin than to quantify the protein, which is at lower concentration. And we call it balanced concentration spiked in internal standards, and then we're really pushing the percent CV to 2 to 4%. and all of them are below 15%, and this is well what FDA would like to see. So really the internal standards are really quite important to improve the reproducibility. Another thing is like, if you have internal standards, it's a pretty busy slide, and I don't want to go into too many details, but what it really helps is like to detect interferences, chemical interferences, because these ions, what you're getting, the ratios of these ions, they need to be identical if you are looking at buffer versus plasma. The internal standard ratio from the Y5 ion, from this peptide over here, which is like 1, 2, 3, 4, 5, this here, and needs to be identical to the Y4 ion, which is 0.22 in buffer as well in plasma. You see, like 0.22. And this should be identical to the endogenous, which is the same, 0.22. Now you can be quite certain that you don't have any interferences. You never can say you have absolute specificity, but using these molecular parameters and characteristics like the mass of the peptide, the mass of the fragment, the retention time, and as well the ratio of these ions, you have four characteristics, they're all orthogonal to each other. We can actually say like we have pretty good specificity over here. And actually, if you have like two peptides, you have two peptides per protein, and then that gives you like ideally like the same quantitative result. Again, like we are using peptides for quantifying proteins. So, but not necessarily they're giving you the same results, since there is always what you need to consider is that you're making the peptides by proteolysis. So you cut them, and sometimes trypsin is not efficient in the same way at every cleavage site. So what can happen is that we sometimes the cleavage is somehow blocked or hindered. But it doesn't really matter. It's very precise. And this is really the point. The MIM using internal standards is a very precise method. So if you plot then the two peptides against each other, they need to correlate quite well. In this case, it's 0.95. And if they are, if you're running then plasmas, running then patient samples, and if it's not, it's like something is not strict on the line, then we can say like this patient over here and this patient we have some interference. And this is quite unique to this approach that we have here, and a technology which can determine patient-specific interferences. And next thing is, if we really want to bring it into the clinic, what we need to have is some kind of automation. And you can do this entire sample preparation shown over here. This can be almost done in a fully automated way using a liquid handling system. And then we really can analyze hundreds of samples in a day. Our largest proteomics project so far was 3,600 plasma samples. And we have done this within a couple of weeks, but it wouldn't be possible at all if you have to do it by hand. This really increased our throughput, and also since no pipetting mistakes are involved, it certainly improves our reproducibility. So where are we right now with this technology? And what we think is like we can quantify disease-related protein biomarkers in human plasma. Is one of our focuses, what we have done in the last couple of years. Here is the general approach, how you do this. You're starting with the plasma, and then you always have the debate, should I deplete the most abundant proteins or should I enrich it? And then you need to have an internal standard. You can add the protein or you can add the peptide, as we are doing. So first thing, like we don't believe in depletion, and we have shown it over and over again. If you deplete, you improve, you get more variabilities, and your reproducibility really goes down. We also have shown that when you deplete, it not only depletes the most abundant proteins, you also deplete other proteins up to a factor of 50. So I really think depletion is not what we should use. Like I make a big point out of it, like do not deplete. Then we don't have internal standards, protein standards. We have using peptides because I'm a chemist. I know FMAR chemistry. I can make my standards, and I'm making that around like 50, 60 standards every week. Then we added to the digest the standards, and then we concentrate and dissolve, and then we can do either directly. We can go straight into our LC-MS system, or what we can do is like we can pre-fractionate this, and then going from there into our LC-MS system. So this is what we're getting out right now. If we're using our one-dimensional LC-MS, we can we can quantify around like 150 proteins, and they cover a concentration range from the milligram per mil albumin all the way down to around like 40 nanograms per mil in this case. And these proteins are, in most of the cases, are being quantified using more than one peptide, and some of them even like up to 6 peptides. The more, the better. And we wanna push this even further, that we have like in the end that all of the proteins are quantified at least by 2 peptides. And then when you see like the distribution, are these peptides of interest? Like, are they somehow related to diseases? And it's like, yes, like 36 of all these proteins which are in here, they are somehow related, according to the literature, to CVD, and another 36 to cancer, and so on. I also like just look at the performances, analytical performance of this approach. It's pretty robust. The retention time is right on the money. We're not using nanoflow. We're using the normal regular flow, because we have, our idea was like, well, nanoflow is quite good. It's very sensitive, but and it makes sense if you don't have a lot of material. If you do a nanoflow system and analyze the plasma, only 14 nanoliters of plasma are going onto the column. But our idea was like, oh, well, the nanoflow is never goes into the clinic. It's way too complicated. Why not going to regular flow rate? But having a bigger column, adding 10 times more, means like instead of 14 nanoliters, 140 nanoliters. Every person can spare 140 nanoliters of blood. And it makes it, the entire system more robust to get in. Then you can use a UPLC. We have very sharp peaks with a width of around 6 seconds in a half peak. So, oops. And then again, like the response is in CV is quite good. The response of the signal is quite stable, and again, the sensitivity, it covers 6 orders of magnitude in concentration range. But we also can do 2-dimensional, what I said, like a pre-fractionation. Here we're using as well HPLC, reversed phase, but we not do this with a low pH, by higher pH. We have a couple of fractions, and now we can actually going almost like I don't know how much, like here in this was like 176 peptides are in here, and right now actually we're almost like we can do like, that's the older version of the slide. We can now do 260 plasma proteins. We can quantify 260 plasma proteins, and we expect at the end of the year we can absolutely quantify around 500 plasma proteins in blood. So we not only can do blood, we also can do CSF and urine. This is what we're just trying to publish. Like in here, we can quantify around like 130 proteins in CSF, and we're expecting to have at the end of the year like something around 250 proteins we can determine in CSF. But we still, I think, I don't know if I mentioned this, like here, like our sensitivity limit with this approach is at this point in time around like 250 picograms per mil. But there are still like a lot of other proteins which are present at a lower concentration. And in this regard, like what we do is we use a combination of immune enrichment and mass spectrometry. And we like to use MODI-MS since MODI is a very robust system, and there are many MODI instruments right now present in the clinic. Again, like we want to bring in mass spectrometry or mass spectrometric quantitative analysis of proteins into the clinic. And it makes total sense if you're building up assays for platforms which are already present. And one of these platforms is a Bruker platform. The Bruker body TOFMS, a little thing. It's like actually the only weighs 35 kilograms. You can take it home if you want to, and you can plug it in. It only needs a plug. That's it. And it has been sold 1,000 times in Europe for microbiology. It actually has changed the way how microbiology is being done, and totally. It's really a disruptive technology. And now we think we want to build this up on this platform. There is a competitor that's a similar platform from Shimadzu. But anyway, like we think like MODI-MS is the way to go. MODI-MS goes into clinic. Both instrumentation from Shimadzu and Bruker are now, like it just got last year the FDA approval, so they will come to the US as well. So here is the scheme of our approach. It is we enrich the protein or the peptide of our interest by using antibodies, anti-peptide antibodies, and then we incubate these immobilized antibodies with our sample, and then we adding, of course, like as the same old trick, the internal standard to it, and then we wash it, and they're placing these beads directly on the MODI target. We're not eluting before, because if you elute something before and it's very, very low concentrated, it sticks. It sticks on the pipette tips. So we're using these beads shown over here and placing them directly on the MODI target. And here are summarized advantages of this technology. So we really can here like enrich of low abundant targets. It's very specific due to mass spectrometry. It's very much an ELISA. You have like a capture antibody, and we have a capture antibody over here. And then in ELISA, you have the detection antibody. And the detection antibody in this case is a mass spectrometer. And then we get like a great specificity because like we can do MS and really gives us a structured information. Yes, it is my peptide of interest because I can sequence it. Minimal sample loss if we really place on the beads on the target and fast results. With a 1,000 hertz laser, you're getting like every few seconds you can measure samples. We calculated one time like you can easily measure 5,000 samples in a day with this kind of system. And and it's quite inexpensive, like a couple of cents for the cost. And it can be totally automated, and it's quite easy to use. And also, we can do it in a multiplex way, probably not 100 proteins at a time, but something in order of 10, 15, that should be feasible. Okay, we have built up now an assay and for the clinic, and it goes into the clinic into the St. Paul's Hospital in Vancouver and the Jewish General Hospital in Montreal. And this assay is for measurement the Renin activity in plasma. Okay, the plasma Renin activity is a measurement for primary hypertension, and this is caused by a dysregulation of the renin, angiotensin, and aldosterone system. And over 1 billion people are suffering from hypertension. And the 10% of them, it's still like 100 million people that are suffering from the primary aldosteronism. Like, and what is it exactly? The angiotensinogen is getting cleaved by renin. It's an enzyme, and it cleaves off the C terminus, the N terminus, and then it releases angiotensin 1. And this has no function, but it's getting further cleaved by ACE to angiotensin 2, and it does all the biology. And if you have the PA, then you're getting less renin, and in the end, it means like you're not producing enough angiotensin 1. And but the good thing is, like, if you can detect it, you can actually cure it, because most of the case it is based on the tumor, it's your adrenal gland, and you take it off and then you are fine. So here is our assay, like plasma activity, how we determine by this immunomodi or short I-modi. We have our plasma sample, and then we split it off in two, and for one sample we incubate at 37 degrees, and it generates the angiotensin 1 through the activity of renin, and our control is on ice, where you also incubate it for an hour, where you have less angiotensin 1 production. And then you're adding the internal standard, same old trick, is an isotopic labeled angiotensin 1 as a cis peptide. And then after this, you place it on the MODI target. But you also produce a calibration curve, and here this is just in PBS buffer, and you add angiotensin 1, the natural as a cis, as a constant, and the natural as a variant, and then you incubate this with an antibody beads, and then you place it on the MODI target after washing. And what you get is these kind of peaks. For every measurement, you get 2 peaks. That's the peak at lower M/Z corresponds to the angiotensin 1, endogenous angiotensin 1, and a couple of M/Z, couple daltons higher in mass, you get the peak for the cis angiotensin 1. We're expecting a much higher concentration of angiotensin 1 after the incubation at 37 degrees compared to 4 degrees. So this is actually what we're measuring. And this is how it looks like. We have like here our standard curve, our calibration curve, and then we have one measurement, and actually we do it in triplicates at 4 degrees and one at 37 degrees. And what we have shown is that the slope of the standard curve and these curves are identical. So we just need to have these 2 measurements, and then we extrapolate this to the x-axis, and the difference from here to there corresponds then to the generation of angiotensin 1. And these are the 2 instruments we think is like well suitable to do this kind of work. And we have here a liquid handling system with small system, like, and what it can do is like it can pipette 96 at a time, so totally compatible with the 96 well plate, and then from there it goes then direct onto the MODI target, and from here into this machine. And so how does it look like? And this machine actually can do both in the reflector mode and in linear mode. These are 2 different modes. The reflector mode is has more higher resolution than the linear mode, but what we're getting like that, what we see is like these reproducibilities is pretty much identical if we're using the reflector mode and linear mode on the intra-day comparison, and as well inter-day comparison, as you can see. So the main question is really, how does it correlate now to the assay, which is currently employed in the clinic. And you won't believe it, but many assays that are really coming out of the 70s, and they're still using radioactive material, including this assay for plasma renin activity, which is a radioimmunoassay, and they're using isotopically radioactively labeled angiotensin. And now we compared these plasma renin activity results with our results, and you can see they correlate quite well. And of an R-square of 0.9392, and reflector mode as well, linear mode. What is very important, the linear mode, because the linear mode is as good as the reflector mode. That means you don't have to, or you can use a lower grade instrument, because the reflector costs more money, and money is like really an issue in the clinical chemistry lab. What is different is the slope over here is by a factor 2 higher, and actually this is how it should be, because we're incubating at pH 6, not at 7, so we have 2 times higher generation of angiotensin 1. So it all fits very well, and we are quite happy about the results. And what I said before, like this assay is going now into the clinic. Before I stop with my first part on quantitative proteomics, I want to talk a little bit about dry blood spots. As you know, dry blood spots, if you're collecting the blood samples on dry blood spots, it has many advantages, which are shown over here. It's less invasive sampling procedure than if someone comes with a needle and sticks it into your arm, at least like for me. Then it's also simpler in initial processing and storage requirements, and I see like people are storing their blood spots just like in a shoebox, and you can store hundreds of blood spots in there. And the sample volume is smaller. You just need more or less a few drops of blood, and this is actually what we do in MIM. We don't need much more, and it is quite stable at room temperature as well. You don't need to have the big freezer, the minus 80 and so on. And something else is like very important. We have to reduce biohazard risk. Like I don't know all of the viruses, like, but at least like HIV and so on, like they're not stable on this one. After half an hour, they're gone, they're dead. So it's quite important. And actually, like using dry bloods for clinical analysis is nothing really new. And actually, you can get this over the counter in Canada. You can buy this when you blood spot test, not for proteomics, for hormones. And we think it's a great idea. And why not using this for the quantitation of proteins as well? And then we said like, okay, let's check it out. And here, like we checked it out like for the 41 high abundant to moderate abundant proteins. And then you would say like, well, it's really of interest to quantify these proteins. Yes, it is. Like actually, it's like most of the proteins what you see over here is actually what the doctor is analyzing when the doctor is doing your blood work. And actually, for all of these proteins in red, this is like these are FDA cleared or approved protein assay targets in plasma. So this is like how the MS looks like. The iron chromatogram, and you see like you have again like the blue lines and the red lines. The red are the internal standards, and the blue is endogenous peptide. And so like that looks like very easy to do, and we checked it like the first out like, so how do we remove then the spot? We can punch it out, shown over here, or we can cut everything out. And so like, let's check the reproducibility, and you can see that on average you have 8.4 here and 5.1 over there. It's a little bit better, but it's very comparable. The next step what we need to do is we need to digest. And we can do this in two different ways. Either on paper digestion, means that we have the paper, we throw it into the digestion solution, or we first we elute the proteins and then we put it into the digestion solution. And again, like we just looked at the reproducibility, is very much identical, so it doesn't really matter. Both works. So quantitation, can we quantify this? Like, and we did a dilution and we get a linear response. So it's definitely quantifiable. That's a good thing. The next thing is like, okay, let's see like the 41 proteins. What do they cover right now? What kind of concentration range can we cover? Around like here from 10 microgram per mil down to around like albumin, it's like milligrams per mil. So around like 4 orders of 3-4 orders of magnitude, I would say we definitely can do this. Another question is, so what are we detecting here? Is it different when you analyze the whole blood versus the dried blood spots? And I was like, actually no, like we all of these 40 proteins or a little more, most of them be detected in both, in dried blood spots and whole blood. And again, like the reproducibility is very similar. And also like the question is, okay, it's very reproducible, but what is like the absolute amount? Is this identical? It should, right? And indeed, like you have a good correlation factor of 0.97, and the slope is nearly 1. So we were quite happy with this, right? So, and we want to improve this, not only having like 40 proteins detected from dried blood spots, ideally a couple hundred proteins. So, and then another question is like, how long can we store it, and what are the best storage conditions for proteins? You know, for hormones, they're quite stable. How is it like with proteins? And we can say like we have like the storage for 10 days versus compared to 1 day, and not all of these proteins are actually stable. We need to check it out, but most of them, they're quite stable within 90 to 100, 110 are most of these proteins in here. And it doesn't really matter if it's 37 degrees, 4 degrees, or minus 20. All look pretty much the same. This is pretty much what I wanted to say, like about, somehow my summary slide is gone for the quantitative proteomics, so I can summarize it just like verbally. I think what I've shown, like what this MIM technology is certainly suitable for quantify hundreds of proteins within one experiment, very reproducible, very precise, not necessarily all the time accurate, but very precise. This is also doable if we're not analyzing plasma samples, also like with CSF, and also like blood samples presented in dried blood spots. So like the last 10 minutes, 15 minutes, I want to talk about structural proteomics. What I think is the next big thing, like in proteomics, like, and I mentioned it like when I talked today, like with the students at lunchtime. So like, where do we go, like in proteomics, and what is the next big thing? I think structural proteomics is for the next couple of years, 10 maybe even more years, we will make also like great progress in this. The same as what we have done like 20 years ago when the word proteomics was invented at the Siena conference, and exactly 20 years ago. And then like the main focus was on protein identification, and now we have a good handle on protein quantitation. In the next 5 years, everyone can do it quite well. And then after this, we're using MS-based technologies to determine the structure of proteins. And here's like what we have done so far. And we use our structural proteomics approaches we developed to analyze or to get some structural information of prion. As you know, the prion phenomenon is that you have this totally native and good prion. It's totally fine, and all of a sudden it converts itself to something really, really bad, and we don't know exactly how the structure looks like after the conversion, and then it starts to aggregate. We have like this fibril structure, which is insoluble, and it's very robust, and it's actually you can burn it, like it's still stable, quite amazing. And then we have this beta oligomers. It's still soluble, so we can use it to study, but it's very cytotoxic, and both are then very beta rich. This is what we know. So what has been done in the past is like the classical structural biology approaches, NMR and X-ray, try to analyze the structure for both, like for the aggregate prions and the converted one, but we only had limited success so far. So our approach is now to use cross-linking as one of the approach for to analyze the structure and the conformational change of the protein, structure of the oligomers, and the conformational change. And we have used a bunch of different cross-linkers, and some of them they vary in the space length, and it's very important for talking to computational scientists that we want to have like very small cross-linkers, so that we have like more restrictive constraints. And this is in the end what we give the computational scientists, and like they are so close together. Like, and if we say exactly they are together like this, and then we have less constraints than if they are further out. Yeah. And different reactivities, and this is all what we have done in the past. So the only problem what we had here is like, well, what we want to do is like we want to convert this into, we want to determine the structure of the monomer after the conversion, but then it starts to aggregate, and as well we want to know what the structure of the aggregates are. But if we do the cross-linking, we don't really know. We cannot really distinguish, since the prion is exactly the same. What is it? Is it an intra cross-links or an inter cross-link? And we have seen all the cross-linkers we have used, and if you put it into the structure of the prion from the native one, which is has been solved, which is like here. It's a structured C-terminal domain from 90 to 232. This is known, and we're getting up this mess because we cannot distinguish between intra or inter, because they're identical. So, but what is known also is like, or what is hypothesis is like this beta 1 H1 beta 2 loop is involved in the conversion. I think it goes, it opens up, and then it leads to the aggregation. So and we came up to, we looked at this paper from John Taverner, 2002, and he used N15 labeled protein and mixed it together with N14 labeled protein. So in our case, if you have seen the prion, one is N50 labeled, one is N14 labeled. If we have now intra crosslinks, we have either this, like we're getting two greens or two blues. But if we have inter-crosslinking between two proteins, we're getting this quadruplet. And so we want to, if we're seeing like a more doublet, then we can use it for determine the conformation changes. And if we see this kind of ion signature, then we can use it to determine or get structural information of the inter-subunit contacts. So I want to explain in a little bit more detail. So again, we have one set of N14 labeled one and the other one N15 labeled one. If we're building up now the aggregation, and here's like the starting with the aggregation is a dimer, we have the following possibilities. We have green, so like two N14 prions. We have one N14, one N15, one N15, one N14, and two N15. So here we cross-linking, then we digest, and then we have like the two peptides which are then connected through the cross-linker. One peptide contains 3 nitrogens, the other one 13 nitrogens. Now, if you compare the same peptide over here at the same crossing to the N15 one, then we have a mass shift of 16 because this is a number of nitrogens. So, and indeed we do see this here in our spectrum. Here we labeled we crossing this protein with a 0 length crosslinker EDC, and here we have crossing the lysyl residue 101 and this E211. And here we're seeing exactly a doubled 16 Daltons apart, and this corresponds to these cross-linked peptides. One has 3 nitrogens and the other one has 6. Now, no, that was wrong. It was 3 and 13, yes. Now we're coming to an example where we have an inter-protein crossings. We have the same possible formations, the green, blue, green, blue, blue, blue. Now, in this case, we're having, for example, like we're crossing in the peptides, one of them has 6 nitrogens, one has 7 nitrogens. If we're going from this formation to that one over here, then we have a shift of 6 Daltons because now the upper one has 6 nitrogen, has 6 and 15 in there. And this is what we see over here. From here, this has then 7, we see a shift to 7. And if we have blue, blue, then we have 13. And this is the example shown over here. And here we have like an intercrossing, and which was crossing with pickup, with crossing tyrosine residues. The structure shown over here. And we have these quadruplet. One is like 6 Daltons apart, other one 7 Daltons apart, and the last one 13 Daltons apart from the N14 labeled crossings. So it looks like quite complicated, but actually it is not really complicated. It's just like simple math. And if it's simple math, you can put it into a software package and you can fully automate this. And this is what we did, and we wrote our own program, and which can analyze this in a complete automated way, looks for this specific signature in the mass spectrometer, and analyzes and assigns then the MS spectrum. And actually, it's really very helpful to have these N15 labeled because it helps us to assign the fragmentation. It's shown over here. Here we have 22 nitrogens in there, and here we have 4 nitrogens in there. And this is like these doublets, what we have in the MS fraction. And this is a B ion 9, which is looks like over here, from here to this, from here to this one here. And it's 16 Daltons apart. And the B10 will be 7 Daltons apart, B11, 18 Daltons apart, B12, 19 Daltons apart, B13, 20 Daltons apart. It really helps us like to assign the fragmentation. It also does like when we said like 0 length cross-linking. It's the greatest, it's the best thing for the structural model list. since we have the highest constraint. But it's very difficult to find them because normally if we're using a cross-linker, which is isotopic coded, it gives us always like this signature. And here, like this cross-linker has, we have two derivatives, one with hydrogen, one with deuterium. So we have a doublet, which is 8 Dalton apart, so we have like 8 hydrogens or deuterium. We immediately see it. But if it's zero length cross-linker, the cross-linker is gone. Just initiate the cross-linker, then it's gone. So how can you see it? But if one is N15 and N14 labeled, then we have this doublet. So it really helps us to find this. Okay, now to the results. So we have here with the intra-protein crosslinks, we found all these crosslinks in prion, and this is like how it looks like. And we have made actually a short movie, what we think. And I don't know how to start it. So here, what the hypothesis was like that the beta 1, H1, and beta 2 is involved in it. It opens up, and then what we think that the N-terminus goes in there. And this fits very well with our cross-linking results as shown over here. It really goes over. Here are these constraints, the distance constraints, and now finally all of them, they're fitting in to having this kind of structure. But it really confirms what the hypothesis is, that this interface opens up, and then the N-terminus actually goes in. But again, like this is not, it's just a model, So, and actually this site has been confirmed not only with cross-linking, I don't have the time, with other structural proteomics approaches like limited proteolysis, like hydrogen deuterium exchange. It's a very powerful method. And as well as surface modifications, where we differential labels this, like the native one versus the pre and after the conformation. And all pinpoints down to the beta 1, H1, H2 section. And we have some results from the interprotein crosslinks, like how do they stack together, the two prions. And we put this also into a movie shown over here. So first you see again like the conformation of each of the proteins, and then the distance are now the match with our results. And now these proteins, the two prions are coming together. And then it is like, we think it's stacked like this. It's kind of a screwdriver on the top. And I have a better picture, which is shown over here. So this is what we think right now, what the structure is. And we collaborating with a very famous structural molecular biologist, and actually this person actually confirmed like this structure so far. So, but we still have like a lot of stuff to do like this over here. What we think is all like beta cheat regions. It's not defined through our experiments so far. So I think here I have a summary slide. Yes, we showed that limited proteolysis indicates increased accessibility for pepsine. I didn't show this actually 150 or 158 region in the oligomers, surface modification, and also that shows that this number of residues in the beta 1, H1, beta 2 loop is involved, increased accessibility. HTX pinpoints the same region where we have like the action, and as well the cross-linking confirms this. And all of these things is like they're all pinpointing down to the rearrangement of this beta 1 H1 beta 2 loop in this oligomer. I only have like the acknowledgement for the structural proteomics people. Like two slides are missing. I don't know what's going But here, like for the structural proteomics part, is mainly responsible Jenna Petrochenko, and with Jason, a student and a technician, and David Wishart from the University of Edmonton, who helps us like to confirm our models and by modeling. And Daryl helped us like with the MS partner, and of course funding. Genome Canada and Genome BC, Pioneer Canada, unfortunately it's not there anymore, and Creative Molecules, and I would like to thank you for your attention. Questions?
Yes, do we have questions for Christoph? Start here, maybe?
Yeah, that's a very interesting talk. So when you do this cross-link, how much or whether it's possible you put the cross-link, your peptide will influence the structure which the targeted protein of the structure.
Yeah.
Because probably you will induce some conformational change by itself.
Oh, okay. Yes. This is always possible, and this question is totally legitimate. Like when you do the crossing, you might change the structure of the protein. That's right. But that's the reason why we're not only using one structural approach. We're using like the HGX, and all of them in this case really show that this region is involved. But you're absolutely right. It is possible when you're doing some kind of protein chemistry that you're changing the structure.
Is there any way to avoid that?
Yeah, we need to be careful. Like you cannot over titrate, like over cross-linking this. If you, for example, we check it out with simple SS page. If you want to cross-link so like a monomer to a dimer, and you know exactly the structure is a dimer, and you see it like on a nice SDS page, okay, adding the crossing at certain kind of concentration, I have now the dimer. If it starts then like truly to aggregate, then you say like, oh no, you overdid this, you put too much crossing in there.
A second very quick question is, in your movie, the model, you go from a very long range distance to the shorter. So how you get the long range distance?
It's just a movie, just so to show like what might can happen if it moves around. In the end, it's like we don't know. It's just like this is like a way to understand this how it moves around,
So
but nothing to do. It's just like a few pictures. But in the end, the model what we have, this fits with the data. That's the only thing.
Yeah, this is a more general question relating to MRMs. There are certain clinical biochemists, including one at Toronto, who still very much believe in ELISA assays. I'm a great believer in MRMs, but what they claim is in terms of sensitivity and reproducibility and producing large numbers of samples, you just can't beat, assays. What do you say to those people?
Die out. That's like some people are a believer, and like, but I actually when I met quite a lot of like clinical chemists, director of the clinical chemistry lab, and most of them they want to go away from ELISA, and but some of them they're hardcore believer, and you cannot convince them.
Yeah, I had to listen to this argument at a previous EDRN meeting where they were discussing various assays that they were trying to develop using MRMs, and this particular clinical biochemist is very influential. Just wouldn't believe it. I mean, that's the bottom line. So,
it probably has some kind of stock, like in ELISA companies or whatever. Like, so it's but in the end, the good thing is like you obviously have a non-believer, but in the end, the science will win, no matter what. And ELISA has problems with specificity, and ELISA has. I also like to develop an ELISA, and the clinical ELISA is like $4 million.
Yeah. But there's certain ones that are very well developed with well validated
Yeah.
antibodies. This is the argument they're going to give you, right?
Yeah, yeah. Well, like, we're not there like for. I don't see like that MRM will totally replace the ELISA. There's some ELISAs are great, and you cannot achieve sensitivity. That's totally fine. But what I see is like what a lot of pharma company, what they need is like really access to fast development of these protein assays. FDA says like no new drug without a biomarker. Like they need to show that the drug is working, and they can companion. It's called companion diagnostic, and this is MIM. We can develop this in quite fast, like within a couple of weeks. It is there. We're synthesizing the peptide, we're optimizing the conditions, and then we show that we can detect the endogenous peptide. And here we go, and then you can
I
run.
agree. In many cases, you just don't have a good antibody or well-developed
Yeah.
antibody, right?
Yeah.
But yeah, there's just certain people are hanging on. I guess the other issue here, we discussed this briefly, was what happens in the case where you're looking at specific changes in like glycosylations or phosphorylations, and the assays really need to depend upon that. The real problem is how to develop an ELISA or multi-immune ELISA.
So
How
what
do
we did
deal
like
with that?
this phosphorylation, we actually went and this is published in the chemistry, we went like the opposite way. We determine the phosphorylation by looking at the non-phosphorylated peptide before and after dephosphorylation. And with glycans, you could do this as well, but only you can determine what's the amount of glycosylation, but you don't know the structure.
The structures, yeah.
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 But if you do with PNGSF, like you throw it in and then it converts it, like the end glycans converts it and get rid of it and converts like into D with a mass shift of 1 Dalton. I don't know, it's tricky. But with phosphorylation, that works quite well. So we determine just the concentration of the non-phosphorylated peptide. And then we do it again after dephosphorylation and also in the jumping. And the difference is exactly the stoichiometry of the phosphorylation. And we can do it with MIM.
Good. Okay. Thank you.
You're welcome.
Do we have any other questions up here? Okay. Maybe I'll just ask a brief technical question. I wasn't sure for your MRM, how many product ions are you looking at? Just one?
No, we're looking normally 2 qualifier and 1 quantifier.
Okay.
But in the end, it's like you have done it over and over again. Why should you look at the qualifier again?
Yeah, but at least you do that first.
Yeah, in the end, like, you know what, we have run it like 100 times. Like, and you're using the qualifier, actually, only the qualifier to quantitate, right?
Yeah.
So you get rid of it. And the better, the less condition you have, the better it is. And the more multiplexing you can do.
All right. If there are no other questions, let's thank Christoph again.
Thank you.
Thank you. Nice job.